When we look at the individual diseases, specifically in Chapter 16, the diseases and infections that are associated with the skin, the integument, or the next couple of chapters, the nervous system, cardiovascular, whatever. For each infection, each disease, I want you to keep in mind these 6 criteria, these 6 things that, we're going to try to break each of these down into, so that way we have some normalcy going from infection to infection, disease to disease. You know, what are the characteristics of this infection, this disease? What are the normal signs and symptoms that, from a clinician standpoint, what you look for? How is this disease transmitted? And the epidemiology of it means who's actually getting infected, who's getting exposed, who's actually getting sick. The pathogenesis and virulence factor, we're going to be looking at what's causing the severity of the disease. Which virulence factors? Is there some enzyme, some mechanism that's allowing this bacteria, this virus, to do more damage, which makes the disease more pathogenic, more whatever? 5 is the infectious diseases lab portion. How do you go through and identify? What are the special things about culturing it? What are the special things about diagnosing it? And then finally, treatment and prevention. What are the best treatments? Here's, the best antibiotics. Here's the best antivirals. Here's the best pills, the best creams. So the first one we're going to start off with is MRSA. Methicillin-resistant Staphylococcus aureus. Okay, this is now becoming a bit of a bane in the hospital setting due to the fact that, well, it's resistant to many of our antibiotics we have on the market. Methicillin-resistant Staphylococcus aureus, MRSA, is segueing into what is referred to as VRSA, VRSA, vancomycin resistant. Since methicillin-resistant Staphylococcus aureus is resistant to pretty much all antibiotics, and our last great hope is vancomycin. Well, we're finding strains, substrains that are now becoming vancomycin resistant. So we're in a bit of a worry. Keep in mind, Staphylococcus aureus, gram-positive cocci. These are considered to be highly virulent. Okay, even though it is part of our normal flora, you're going to find it on your skin. You're going to find it for some people, in their throat. It's everywhere. It likes being on the skin. It likes the salinity. It likes the low pH. Does very well. The issue is that it's able to penetrate sometimes. Microabrasions in the skin, scratches, buildup of sebum blocks hair follicles, blocks oil production. Well, in that blocked hair follicle, in that blocked oil gland, it's going to be different bacteria from the surface of the skin. One of them is going to be Staphylococcus aureus, as you see here in this picture. It's going to be a large raised lesion. Okay, it's not a pimple. This is going to be much, much bigger than just a pimple. It's going to be highly inflamed. You see that red ring around the lesion. Lots of inflammation. It's going to be very warm to the touch. it's going to be fluid filled. And the fluid, it's not going to be clear fluid, it's going to be pus. So you give it a squeeze, you scratch at it. In a clinical setting, you may lance it. Out's going to come pus. The transmission here, well, anytime you touch stuff that somebody else has touched, you're going to get transient microbes. Those transient microbes are going to include new strains of Staphylococcus aureus. What makes this MRSA an issue? Well, turns out under normal conditions, Staphylococcus aureus has a wide range of virulence factors, weapons at its disposal. Things like coagulase. Coagulase is an enzyme that coagulates blood. So blood flows into an area, but because all the blood cells coagulate, they form giant clumps. They get caught up in the area of the infection. Blood flows in, blood doesn't flow out. Everything begins to pool. Neighboring cells die. Your cells die, they burst open. Basically, all the intracellular contents now become available to the bacteria causing the infection. It now becomes new nutrient sources. Also have DNases, lipases, different things that are going to go through and lyse neighboring cells. Culture, well, blood agar, mannitol salt agar. Blood agar, remember, normal agar has sheep's red blood cells in it. Turns out Staphylococcus aureus, one of its virulence factors is an enzyme that causes red blood cells to lyse, burst open. So you'll be able to see this on the red blood agar because instead of being fire engine red, it's going to have zones where all the cells have been lysed, so it's now clear. Now, Staphylococcus aureus is not the only gram-positive cocci that has lysis capabilities, but it is the only gram-positive cocci that's going to be in clusters that is catalase positive. Catalase is an enzyme that takes hydrogen peroxide, H2O2, which can be damaging to proteins and DNA, takes H2O2 and turns it into water, H2O, and gaseous oxygen. So hydrogen peroxide, the stuff that mommy used to pour on your cuts, and then it would bubble. That's bubbling because of catalase. Staphylococcus aureus in the cuts, in the scrapes, turning the hydrogen peroxide into water and oxygen, two things that are not damaging. Prevention, well, basically it's just good hygiene. Staphylococcus aureus is part of our normal flora. It's everywhere on us. Best thing to do, wash your hands. Take a shower every other day or so. Use soap. Clean clothes. Now, once you do have a staph infection, gonna need to lyse the lesion, get the pus out, drain the pus. If you do not do this, that lesion will begin to spread, increase in size, and then we end up with the wound infection we associate nosocomially. Okay? MRSA in the hospital is literally nothing more than staph infection of an open wound. It becomes one giant lesion, pus-filled. That giant lesion will not heal. So it will be a continued source or continued site for further secondary, tertiary bacterial and fungal infections. Because Staphylococcus aureus is becoming more and more antibiotic resistant, they like to use antibiotic cocktails now. It may be slightly resistant to antibiotic A, but not so much antibiotic B. So you, instead of one or the other, you come in with a 1-2 punch. A knocks down the numbers, and what A can't get rid of, you hope that B, which is being taken at the same time, B will take those numbers down even further. So you may not be able to wipe out the infection chemically, but you can keep the numbers down low enough that hopefully in the next few days, the patient's immune system will catch up and will remove. Here in the States, because MRSA is spreading through our hospitals, it's become a bit of an issue. Part of that chemical cocktail, that antibiotic cocktail, will include vancomycin.